I'm going to talk about the comparative regulomics in flatfish. What we tried to do here was to find conserved regularity regions in flatfish based on turbot data. No. Not working.
Try that again now. Is it working?
Oh, there.
Okay. Sorry.
So we started with a list of around 30,000 enhancers located in TURBOT by the analysis of the chip and attack seek of the depth map and body map experiments that Paulino talks about this morning. And as an initial approach with these sequences, we did a blast search using the turbot enhancers and indexed genomes of Cynoglossus semilaevis, Paralictis olivaceus, Hypoglossus, and Solea senegalensis. We used these genomes because there are those that are available on Ensemble Lab today. The output we got was the best single alignment for each turbot enhancer in the target genomes. With this data, what we first did was a filter based on the alignment length. This filter produced lower number of alignments when these alignments were increased in length, being the lowest number of alignments when the 5 species were considered together, followed by 4 and 3 as expected. When each species were considered by itself, Cynoglossus produced the least number of alignments, followed by Solea, Paralictis, and Hypoglossus for all, for all the filters applied. And initially, as a first approach, we analyzed the most stringent filter, which is those alignments that were 350 bases and that were shared by the 5 species selected for the study. This complied about 1,000 enhancers in each species. We initially checked the synteny, and we were able to reproduce the synteny obtained by orthologous genes after genome assembly for each species with the positions of each enhancer in the different species analyzed. For example, chromosome 9 of turbot correspond to the sexual chromosome of Cynoglossus, 12 to the number 2, and so on. This happened also in Hypoglossus, Paralictis, and Solea. The synteny was perfectly conserved, at least for these 1,000 enhancers. Then, to check what was around these enhancers, we selected about 100,000 bases around each of the enhancers for each species and identified the genes in these locations. With those, which were approximately between 2,900 and almost 4,000 proteins in each species, we performed functional enrichment, and we observed that about 35% of the genes around these regions coded for development, morphogenesis, differentiation, and patterning, with a lot of tissues and organs represented by in these terms. This is GO enrichment. The rest of the enriched terms corresponded to gene expression, regulation, and general metabolisms and cell death regulation. At the level of the pathways enriched, a lot of metabolic genes are present around these conserved enhancers, which are mostly involved in general lipid, protein, and carbohydrate metabolisms. Also, the circadian entrainment was enriched, and importantly, these three pathways controlling development, morphology, and organ size were highly enriched. Most in detail, the IPOL signaling pathway, which controls organ size, embryogenesis, and tissue regenerations, was highly covered by enriched genes, being the species with more enriched genes, Cynoglossus, and in this sense, the enrichment of proteins decreased, of genes decreased. For the WNT signaling pathways, again, both the canonic and non-canonic pathways were heavily covered by enriched genes, and the order is exactly the same as the previous case. And finally, mTOR signaling pathway, again, is heavily enriched by proteins in all the species selected except for Hypoglossus. No explanation for this. After genome functional enrichment in the genes around the enhancers, what we did was to look for no motifs in these enhancers. Using Homer, we obtained around between 16 and 80 motifs in the 1,000 enhancers in each of these species, which were recognized by around 30 to 50 transcriptional factors. To compare with the case of the genes, we also enriched the transcriptional factors identified using gene ontology and different pathways enrichment analysis. We observed a similar pattern. In this case, almost more than half of the differential biological process by gene ontology were related to developmental stages, differentiation, and morphogenesis, while the other half was, as expected, related to control of RNA synthesis, transcription, and so on. Again, a lot of tissues and organs were represented among the developmental enriched terms. In the case of the enrichment pathways, we detected only 2 pathways enriched for the 5 species selected, which are developmental biology, myogenesis, and dopaminergic neurogenesis, while other pathways were enriched, but only for 3 species or 2. We then selected those transcription factors that were present and enriched in the 5 species, reproduced the enrichment analysis with similar results. In red are those that are also seen in this case. In black are not seen in this case, but in the individual analysis. And again, the same terms that appeared enriched in individual analysis appeared enriched in this enrichment pathway. Finally, as another approach to identify regularity sequences in flatfish, we use cactus. What we did was perform an alignment of the same 5 genomes. In this case, we included Orysia latipes as an outgroup, and to test if the results of the cactus analysis coincided with that of BLAST. With the previous results, what we did was imputing to the alignment the enhancers identified in TURBOT, and And this is a simple example of the genome browser. What we got out was the positions and of the enhancers located in the rest of the genomes. I mean, in Gynoglossus, Paralictis, Hippolossus, and Cynoglossus. And here you can see that when you, in the genome browser, put those positions against each of the corresponding genomes, again, the synteny is conserved. For example, the number chromosome 9 in Scophthalmus is chromosome 4 in Paralictis, 5 in Scophthalmus is 3 in Paralictis, in Hipoglossus, 3. I mean, the same results appears. We just started with this approach of cactus. We started with this work 2 months ago. We didn't have much time to work with this data. But as preliminary conclusion, we can say that approximately 1 enhancers that are very conserved across flatfishes harbor around 50 to 20% of the protein coding genes in this species. The synteny of selected enhancers recreates that observed after chromosome assembly for selected species. Syntenic analysis of regulatory sequences could be applied for genomic regions, gene markers, and gene markers, that doesn't go there. That goes there. And are associated with productive traits across flatfish. The genes controlled by studied enhancers are mostly involved in development, morphology, patterning, and general metabolisms, and this is supported by enrichment of transcription factor identified after motif enrichment analysis. This is the first stage of this work. Now, this has to be expanded, reducing the stringency to analyze enhancers with lower alignment length and those in less than 5 species, repeating all the enrichment and motif identification in these species, and continue with the cactus analysis. So in the end, to compare strategies followed by, obtained by both approaches. And that's it.